This video will zoom in on M phase in the cell cycle and outline the steps of mitosis. Before we look at the steps in mitosis, let's briefly examine the mitotic spindle apparatus. These are the structures that organize and sort chromosomes during mitosis, and their function is crucial to proper cell division. In animals, the spindle apparatus is organized from the centrosome, each of which contains two centrioles. This works a bit differently in plants. There are three types of microtubules, each of which has a slightly different function. Aster microtubules help position the spindle relative to the cell membrane. Polar microtubules project towards the middle of the cell, and at anaphase begin to push the spindle poles away from each other. Lastly, the kinetochore microtubules attach the spindle pole to the kinetochore proteins at the centromeres of each chromosome. These will eventually pull individual sister chromatids towards each pole, separating them into different cells by the end of mitosis. Mitosis is composed of 5 steps, and many of us have seen this since junior high, so in this video we'll briefly go through what is happening at each of these steps. Note that in the following diagrams we will see a cell with 6 chromosomes for a diploid cell. Interphase represents the steps leading up to M phase that were discussed in a previous video. Go watch those if you don't recall what happens here. In prophase, we see the nuclear envelope dissolve and the chromosomes reach their final condensed form. The mitotic spindle begins to form in this step and stretch across the cell. Prometaphase is represented by the arrival of the centrosomes at opposite poles of the cell and the kinetochore microtubules binding to the centromeres of each chromosome. At this point, the full mitotic spindle apparatus has formed. Metaphase is the easiest of the steps to recognize. In mitosis, this step is represented by a single file alignment of the chromosomes along the equator of the cell, which is called the metaphase plate. Anaphase, the kinetochore microtubules shorten and separate sister chromatids from each other. They are then pulled towards opposite centrosomes of the cell. Last step is telophase, represented by the completed migration of the chromosomes to opposite poles of the cells, followed by cytokinesis, which evenly divides the cytoplasm and organelles, turning our single cell into 2 identical daughter cells. The results here are 2 daughter cells that are genetically identical. They are identical because in the process of mitosis, we separated sister chromatids, which were formed during S phase when our chromosomes were replicated, and they are thus identical. Mitosis thus ensures genetic consistency from one cell to the next, and is the reason why we, as humans, have trillions of identical cells in our body. Mitosis is the process that makes multicellularity possible.